Hello, good morning. Myself, I am BSR, botany lecturer. Today, our topic meiosis. So, meiosis first reported by August Weismann, later on proved by Strasburger in plants. The term meiosis was coined by Farmer and Moore. So, meiosis mainly two steps are there: meiosis one, meiosis two. In the meiosis one, the diploid mother cell undergo division to form two haploid cells. In the two haploid cells, it means the chromosome number reduced to half. Suppose 2n equal to 4, in the daughter cells n equal to 2. Later on meiosis 2, these two haploid cells divided into four haploid cells. So the meiosis 1 is reductional division, meiosis 2 is equational division. So directly we are going to meiosis 1. In the meiosis 1, four steps are there: prophase 1, metaphase 1, anaphase 1, and telophase 1. Now prophase 1. Prophase 1 is the prolonged phase. It takes near about 12 to 24 hours. In this prophase 1, there are steps: leptotene, zygotene, pachetene, diplotene, diakinesis. First, leptotene. So in the leptotene, you just observe haplopappus gracilis reproductive cell. There, how many chromosomes are present? Four chromosomes. All these four chromosomes are long thread-like. It appears like a long thread. So long thread-like chromatin material is present. But important thing, before leptotene, before leptotene, their interphase completed. Interphase G1, S, G2 already completed. When S phase completed, there the DNA becomes doubled. It means DNA replication is over. There the DNA content becomes from 2C to 4C. It means each chromosome containing two chromatids. So anyway, in the leptotene, there the chromosomes are appears like a long thread-like chromatid material. They comes at one particular point and appears like a bouquet. That stage is called a bouquet stage. Anyway, next zygotene. Especially in the zygotene, you just remember if 2n equal to 4 chromosomes are present, in that 4 chromosomes, 2 belongs to maternal, 2 belongs to maternal. So 2 belongs to paternal, 2 belongs to maternal. So their homologous paternal and maternal chromosomes attract towards each other to form pairs. To form pairs. That process is called synapses. Each pair is called bivalent. It means if four chromosomes are present, how many bivalents are present? Two bivalents. So this is one bivalent, this is another bivalent. So it means in each bivalent, how many chromosomes? Two chromosomes. In each bivalent, how many chromatids are present? One, two, three, four. So it is a bivalent or tetrad. In one bivalent, two chromosomes, four chromatids are present. Hence it is called tetrad. Anyway, in the zygotene, elongation of nucleolus takes place and initiation of spindle fiber starts in the zygotene. Later on, we go to pachetene. Here in the prophase 1, the most significant phase is the pachetene. Because in the pachetene, the chromatids of the same chromosome is called sister chromatids. Chromatids of the different chromosomes are called non-sister chromatids. These non-sister chromatids exchange their parts mutually. That process is called crossing over. So during the crossing over, the non-sister chromatids breaks by the action of endonuclease. Later, they mutually exchange and get united by the action of ligase. They are called recombinases. So due to crossing over, their X-shaped structure is forms. That X-shaped structure is called chiasmata. So crossing over takes place. That crossing over leads evolution. So that is why the pachetene is the most significant phase. Later on, when you go to diplotene, so after pachetene, crossing over is over. So that is why here these homologous paternal and maternal chromosomes are trying to separate with each other in the diplotene. They are trying to separate with each other due to weakening of synaptic forces. Due to weakening of synaptic forces. But due to the presence of X-shaped chiasmata, due to the presence of X-shaped chiasmata, these homologous paternal and maternal chromosomes do not separate immediately. Why? Due to the presence of X-shaped chiasmata. So that is why in the diplotene, just a little bit condensation takes place. Later on, after diplotene, diakinesis. In the diakinesis, very important point, there the chiasmata moves from their original position. Here, this is the chiasmata, X-shaped chiasmata. The chiasmata is moving towards their ends. So chiasmata is moving towards their ends. So here chiasmata is there, not in X shape. So that chiasmata is present at the terminal position. That process is called terminalization. So terminalization can observe in the diakinesis only. Learn one more point in the diakinesis, the condensation takes place. Like that, at the end of the diakinesis means end of the prophase, there the nucleolus disappears, nuclear membrane also disappears. So that is why all the chromosomes are released into the cytoplasm. This is prophase 1. Now, metaphase 1. You know, all are released into the cytoplasm. No nuclear membrane, no nucleolus, no other cell organelles. Now, in the metaphase 1, what is the step? Chromosomes are arranged on equatorial plane. Here, the bivalents are there. Chromosomes are arranged on equatorial plane. Yes, here spindle fibers are formed, bipolar spindle fibers. Here, one bipolar spindle fibers are attached to the centromere. Attached to the centromere only to the kinetochore. In mitosis, the spindle fibers are attached to the centromere to this side kinetochore and that side kinetochore. But here, this spindle fiber is attached to only one kinetochore. That spindle fiber is attached to only one kinetochore. This kinetochore is free. And one more thing, here these are the chromatid arms clearly present on equatorial plane. The chromatid arms are present on clearly on equatorial plane. That's the most important thing in metaphase 1. So in the metaphase 1, so chromosomes are arranged on equatorial plane, bipolar spindle fibers are formed, they are attached to the centromere. Later on, anaphase 1. In the anaphase 1, there the spindle fibers are exhibiting pressure on centromere, but no centromere division. There is no centromere division. Whereas in anaphase, centromere division takes place. In the anaphase 1, no centromere division. Just the chromosomes, homologous paternal and maternal chromosomes are separated with each other. They are separated with each other. There only disjunction of the chromosomes takes place. Only disjunction. They are not daughter chromosomes. Homologous paternal and maternal chromosomes are separated with each other. That process is called only disjunction. There is no centromere division. So that is why here four chromosomes are moving, two chromosomes are moving towards one pole and two chromosomes are moving towards one pole. It means chromosome number reduced to half. It's the most important thing in the anaphase 1. Okay, so finally these chromosomes are pulling towards their poles. Later on, telophase 1. Telophase 1 is reversible to prophase 1. It means their nucleolus and nuclear membrane disappears. Here nucleus and nucleolus and nuclear membrane reappears. There the chromosomes are condensed. Here chromosomes decondensation and appears like a thread. So that is why at the end of the telophase 1, at the end of the telophase 1, in each cell, how many nuclei are present? Two nuclei, each nuclei in haploid condition because chromosome number reduced to half. So after meiosis 1, meiosis 2. In the meiosis 2, you know prophase 2, metaphase 2, anaphase 2, telophase 2. These are the steps. Anyway, at the end of the telophase 1, each cell containing two haploid nuclei. It means in this haploid nuclei, you observe here two chromosomes, here two chromosomes. So now prophase 2, that long thread-like chromatin material transformed into rod-shaped structures. Their nuclear membrane and nucleolus is present that disappears and all are released into the cytoplasm. But important thing here, each chromosome containing two chromatids because till now no centromere division. Okay, now after prophase 2, metaphase 2. In metaphase, always the chromosomes are arranged on equatorial plane and spindle fibers are formed. So bipolar spindle fibers are forming. Here, how many bipolar spindle apparatus are forming? 1, 2. So in metaphase 2, two bipolar spindle apparatus. In metaphase 1, one bipolar spindle apparatus. Like that in meiosis, how many bipolar spindle apparatus are formed? 3. In meiosis 1, 1. In meiosis 2, 2. So like that, two bipolar spindle apparatus are formed. So metaphase 2 is also over, then anaphase 2. In the anaphase 2, the chromosomes, here spindle fibers are there. These are the bipolar spindle fibers. So these bipolar spindle fibers are exhibiting pressure on centromere. Here they are exhibiting pressure on centromere. So that is why here centromere division takes place. Centromere division takes place in anaphase 2 of meiosis, anaphase of mitosis. So centromere division takes place and finally the daughter chromosomes are bringing towards their poles. And finally telophase 2. Telophase 2 is reversible to prophase 2. There here rod-shaped chromosomes are transformed into long thread-like chromatin material, nucleolus and nuclear membrane reappears. Like that, at the end of the telophase 2, 4 haploid nuclei are formed. 1, 2, 3, 4 haploid nuclei. You just observe in each haploid nuclei, 2 chromosomes only present. Okay, and finally, cytokinesis takes place. Cytokinesis. After cytokinesis, how many cells are formed? 1, 2, 3, 4. This cytokinesis may take place at the end of the telophase 2 or at the end of the telophase 1. Like that, in meiosis, mother cell divided into two daughter cells. Those are haploid cells. Those haploid cells again divided into 4 haploid cells. This is the meiotic division.